You check, Gabby, if there are some students in the attendees?
I already,
Okay, good.
I'm there, so yeah, I try to see.
So, welcome everyone. And so today we're going to talk about anatomy of a microscope. So let's dissect our microscopes. So there are three parts in this lecture. And in the first one, we're going to look at the architectures of different microscope and in the difference between transmitted light and fluorescence. So microscopy is a very vast field. And there is, for example, material imaging is not something we are dealing with in here. And there is, but it's actually a very big field. This is where most of the money of the field is. For example, computer chips are quality controlled with material imaging. And then we have scanning probe microscopy is also another field that we do not deal with. Clearly, you know electron microscopy, there's transmission and scanning. And that's not what we're dealing with either. And then medical imaging, that you probably know as well. And then there is light microscopy for biological research. It's a quite narrow field, let's say, compared to the rest. So within that field, we have transmitted light images. It can also be called bright field. I will explain. And then there is fluorescence microscopy. And for both of them, we can have a sample that is labeled or a sample that is not labeled, but we can anyway take images of what we have. So in fluorescence microscopy, we also have labeled and label-free samples. For example, sample that is autofluorescent or in reflection. These are samples that are not labeled, but that we can anyway image. Transmitted light microscopy can be done on wide field systems and on confocal microscopes. And fluorescence microscopy can also be done on wide field systems, confocal microscopes, and also light sheet microscopes. So light sheet microscopes do not have any transmitted light imaging. So these transmitted versus fluorescent, these are different microscopy techniques. And wide field, confocal, light sheet, these are different types of microscope. And then on top of that, we have different architectures. So a microscope can be inverted, upright, or flat. So I will show some examples. So these are different types of architectures. So I think my lecture here is going to sort of explain all this, and hopefully it makes sense at the end. So one thing that I want to point out immediately is wide field and bright field. So often people may confuse them, they sound a bit the same, but they're actually very different things. So a bright field means that we have a dark object on a bright background. So this is what we do when we use transmitted light imaging. So the background is lighter than the object itself. Whereas wide field means a defocused illumination, as opposed to, for example, point confocal, where the illumination is a point, okay? A wide field means the whole field is illuminated as one. So they sound the same, they're completely different. So just remember bright field, wide field, and I will show you, and maybe it will help you remember. So the fundamental difference between all these microscopes, these different types of microscopy, is the light path. And this is what we're going to focus on today. So how to understand the light path? In some ways, it's very simple. It starts with a light source, and then you have some optical elements, and then you have a sensor. And that's the end of the light path. So it starts and it ends. So let's have a few definitions also so that we speak the same language throughout the course. And these are terms that can be used differently elsewhere, but I have decided that in this course, we're going to call a sensor a light recording device. So what type of sensors do we have? We can have the eyes, detectors, and cameras, okay? Sensors can be used in other ways outside of this course, but in this course, I try to harmonize so we all speak the same language. So sensors are any light recording device, including the eyes. So what about the optical elements? Where, for example, we have lenses, like including the objective. There's lots of lenses in the objective, but there's lenses also outside of the objective in the microscope. We have filters. So what type of filters do we have? we have excitation filters, and because they deal with the excitation light, they are close to the light source. And then we have emission filters, and because they deal with the emission filter, they are called close to the sensor. So that's sort of logical, the place where they are. The optical elements include dichroic mirrors. And so now what are the 4 roles of a dichroic mirror? You guys tell me. Who's going to say?
I can say one if you want.
Please.
So it separates which wavelengths from the light source will go to the sample.
Good. And so it sends the light, the excitation light, right? The excitation light, because it comes from the light source. It sends it to the sample. Very good. Anything else?
That the dichroic mirror avoid that the excitation light come back to the sensor or go to the sensor.
It prevents the excitation light from going to the sensor. Very good. So we have 2 of them.
Allow that the emission light goes the sample through the sensor.
It sends the emission light to the sensor. So now we have 3. So it sends the excitation light to the sample. It sends the emitted light to the sensor, and it prevents the excitation light from going to the sensor. Very good. We have 3. And then there was another one that you saw in the video. That many of you use on your point confocals.
I think it split the emitted light towards different sensors.
Excellent. So if we have several sensors, we often have, but it's not always the case. We will talk about this next week, but we often have a dichroic mirror that splits the emitted light into emitted light, one of each going to each sensor. Very good. Thank you very much. That was it. So we send the excitation light towards the sample. We send the emitted light towards the sensor. We prevent the reflected excitation light from reaching the sensor. And then we could split the emitted light towards 2 sensors. Well done. Good. So the dichroic mirrors are there somewhere between the excitation filters and the emission filters, right? So they're in the middle. So also quite logical. There are pinholes as well. These are also optical elements. But it's only for confocals, right? And then we have the sample, we have the cover slip, we have the mounting medium, which is the medium that you have on your sample. All these things are actually part of the light path. So we have to consider that the sample is part of the microscope, if you want to say, once you have put it on the stage. So in a very simple way, there are only 2 types of microscopes. Those that work without a camera, and these are single point confocals. And those that work with a camera, and that is all the other microscopes. So wide field microscope, so wide field microscope, and many other types of microscopes. So only the single point confocals work without the camera. So we're going to look at this in details. Now, again, a definition. What we call here in this course single point confocal is what most people call a confocal. Okay, or sometimes a point scanning confocal, or sometimes a laser scanning microscope. And so just to make it very, very clear, we are going to talk about single point confocal and multi-point confocal, just to make sure that everybody knows that a spinning disk is also confocal, and that just makes it very clear from the name. So this is the way we harmonize things. Single point confocals is what most of you are actually using in this course this year. And then wide field, so again, it's wide, so this is defocused illumination. This is what sometimes we hear called standard microscope, sometimes we call them fluorescence microscope, which is not logical because the confocal is also a fluorescence microscope, right? Or epifluorescence microscope, or normal microscope, or sometimes you say a stupid microscope. No, it's called a wide field microscope, okay? So we have a single point confocal and a wide field microscope, and sensors is anything that detects light. And now, from now on, we know what we're talking about. So we have microscopes without cameras and we have microscope with cameras. Let's have a look a little bit. So this first lecture we'll have we will cover these 3 parts, and then this will be one more little lecture and one more little lecture. So first we're going to talk about the architecture. So upright, inverted, or flat. So these are 2 types of microscope. This one is upright and this one is inverted. What does that mean? We look where the stage is. So here we see the stage, and then we see where the objectives are. If the objectives are above the stage, it's an upright microscope. If the objectives are below the stage, it's an inverted microscope. And that is the case for most of the microscopes that we use. And we will see why. What is the advantage of one versus another? But they both work very well. So when we look at the stage and the objective, we will always see something on the other side of the stage compared to the objective, and that is the condenser. So the condenser is always opposite the objective compared to the stage, okay? So here we have a light sheet microscope. So light sheets are a little bit different. We have different types of architectures, but you can see here that this is the stage. These are the objectives, because in light sheet we often have several objectives, and you can see that the objectives are above the stage. So this is an upright light sheet microscope, and this one is a little bit different. So this is a flat light sheet. So you can see the objectives are in here. So if we open the whole thing here, the sample is going to go here. If we open the whole thing here and we look from the top, this is what it looks like. The sample would go in here, and then the objectives are like this. And so this is the sample and the objectives are actually at the same level. So this is a flat architecture that exists really only on very special light microscopes and on light sheet systems, put it that way. So the objective here is, so these are upright microscopes. Let's see what they are. So the objective is above the stage and the condenser is below, right? This is what we said before. So why do people use upright microscopes? There's a historical reason. These were the first microscopes. They were designed that way. But also, so some things have sort of stayed behind. But also it's useful if you have a very thick sample and you want to image the top and you can flip. So it's a quite special application. It's also useful if you want to have lots of pipettes and stuff like this and you need a little bit of space in here because you have no condenser. You can put just one objective, whereas the condenser is quite bulky. So if you have the condenser here, it's harder to put all sorts of pipettes. So that's useful in that case as well. So when I say imaging the top of thick samples that cannot be flipped, one typical example is that if you're doing intravital imaging and you have, for example, you're studying the brain of mice and you have like a little window and you do not want to flip the mouse because it's not physiological. So the brain is going to react completely differently if the mouse has its legs up or not. So in this type of cases, then it's better to image from the top. Or if you have like a tissue model that is in liquid and you need to image the top of the tissue, not the bottom. So this type of cases, you need an upright microscope. The problem with upright microscopes is that the sample and the objective are on the same side of the stage, and so it's difficult to move the stage because the objective will bump into the dish. So there's little stage movements if you have a sample that is in a well, for example. If you have a slide, then no problem. So inverted microscopes, so the objectives are below the stage and the condenser is above. It's an inverted microscope. So it's easier to use if you have live samples and you need a small CO2 chamber. You can put it on top of the stage and the objective being below the stage doesn't bump into it. You can also use multi-well plates. It's the same thing. So the objective doesn't bump into that. You can combine it exactly with all sorts of different multi-wells and dishes without any problem. This is the reason why, because it's more versatile. This is why most modern microscopes are actually inverted. Most modern microscopes, except for intravital. But even for the intravital, it's often inverted, so it really depends. In intravital imaging, you can. So intravital means imaging in live animals. Then you can have both applications. But for most of what we're doing in this course, we're better off when inverted. And I think actually there's only one person who has an upright system in the course, if I understood well. So for a fixed sample between a cover slip and a slide, it doesn't matter if you have an upright or inverted, because the main problem is the stage movements. So here in a little comparison side by side, you can find upright and inverted microscopes on any type of microscope, so any wide field confocal or light sheet. You can do it transmitted light and fluorescence imaging. You can have these microscopes fully automated, both upright and inverted. So in the stage movement, Z-stack, different colors, there's no difference at all. You can use fixed or live samples, and you can do all these specialized techniques in here. However, the upright is best to image the top of fixed samples, and the inverted is best for everything else. One of the main issues with having an upright microscope is that you have no hardware autofocus, and we will talk about hardware autofocus later. Whereas on an inverted, you can have a hardware autofocus. It's very useful. So what about the emission light path? Because we spoke about the light path. So what happens with the emission light path? So here we have the stage, the objective, and the condenser for each of them. The emission light path is sample. So from the sample to the sensor, okay? The excitation light path will be from the light source to the sample, but now we're talking about the emission light path, so it's between the sample to the objective and to the sensor. So the light that from the sample that goes into the objective and then finally ends up on the sensor. So one of the tips is that because we go sample, objective, sensor, that means that the objective and the sensor are mostly on the same side. Okay, so in here, for example, you can see the sample is going to be here, the objective is here, and this is the camera. So that's the sensor. So this is the light path. And we can also decide to send the light to the eyepiece if you want to look at the eyepiece. So this is the emission light path of this microscope. And then in here, this is inverted. So the objective is below, so the sample will be here. The light that the objective collects is going to go down, and then it will be sent to this thing, which is a camera, or to the eyepiece. Okay, so this is the emission light path. Very simple. There's only one exception, and that is when we image transmitted light on a single point confocal. So we're going to see this example. So the emission light path is super simple. It is sample, objective, sensor. But it's different with transmitted light when we image transmitted light on the point confocal. So we'll see. So what about the excitation light path now? And that's different. So the excitation light path is different between different systems. So we have a little look, but before that, we're going to go to the microscope architecture and do a little quiz there. So if we have a look here and in Canvas, you guys can go to Canvas and go to this one. And we're going to do it here. It's fine because it's very short. And maybe people online, if they want to do it in their head, they're welcome. Okay, this is this microscope upright, inverted, or flat? And where is the condenser? Is it above or below the stage?
Yes, I'm inverted, and the condenser is above.
Super. Okay, good. Okay, is everybody okay with that? The system is inverted, and that is because the objectives are below the stage, and the condenser is always on the other side. So if the objectives are below the stage, the condenser are above. Is everybody okay with that? Good. Then I continue. There. So now we spoke about upright, inverted, and flat architectures. Now we're going to talk about the microscope and the satellite equipment. So I tend, and many people tend to call this type of pile of equipment, organized pile of equipment, a system. And that is because we have a microscope body and satellite equipment, lots of satellite equipment. When I tell people that I work in a microscopy facility, they think that we only have this. And they do not realize that it's only a small part of the system. So here, this is the microscope body, and here is all the satellite equipment. That's a lot more. So these satellite equipments are attached to the microscope body with cables, optical fibers, and hard connections. So let's have a look a little bit closer to this particular microscope. And you can see that they are cables, so they look like computer cables, thin or thick. And then we also have fibers, so they look like they have some sort of a metal. They are metal actually, and they're a bit bendy. You have to be really careful with them. Don't bend them. So they need to be gentle with them. So these are optical fibers, and then we have all these hard things there. Like here, this is attached to the microscope directly. So these are hard connections. Oh no, she wants us to learn all this about our microscope. Okay, it's not exactly true, because we're only going to look at the equipment that is attached to your microscope with hard connections. So it's going to be significantly less than the whole thing. And one of the tips is that the sensors, that one tip that we saw before, the sensors are mostly on the same side as the objective. So I think for all of you, the sensors that you have, cameras or detectors, they're going to be on the same side of the objective. So it's quite easy to find them. Because you took pictures of your microscope, and now you're going to have to tell me what everything is on the pictures. That's one of the assignments. You're going to write on the pictures what is what. So we're going to do all this in the anatomy of a microscope later today, the workshop. So we have satellite and satellite equipment, which is attached to hard connections, and there are not many different things that can be attached. So there are cameras, scanners, and we have detectors, a few things. So, so I remind you that we have only two types of microscopes, those that do not work with a camera. Some of them have cannot take images, but we don't care about those in this course. So the microscopes that do not have a camera and can do some imaging, they are single point confocals. And the second type, so this is what people call confocal, but we call them single point confocals. And the second type of system is systems that work with a camera, and that is all the other microscopes. If we have a camera, we could have no scanner, in which case it's the simplest system, it's a wide field system, okay? Or we could have a scanner, and in this case, we have a multiple confocal. So this is what other people call a spinning disk, but in this course, we're going to call them multiple confocals to clearly realize that they are confocals. And there are a few more, but more or less it's going to be this. Do you have a camera or not? And if you have a camera, do you have a scanner or not? So it's not super complicated. So we're going to learn how to recognize a camera, how to recognize a scanner. This is for this afternoon. And then this way you will be able to identify, by just looking at a microscope, any microscope that you have never seen before, you will be able to identify if it's a wide field system, or if it's a single point confocal, or if it's a multi-point confocal. Okay, so good. So that is the fun for this afternoon. So now we're going to talk about transmitted light and fluorescence, what the difference is. And so I spoke before about bright field. So then you know that a wide field microscope is a microscope that has a camera and no scanner. So it's a simpler microscope. It's sort of the simplest type. But bright field, what is that? So it's the objects in the image appear dark on a bright background. So this, for example, are the beautiful images of Waldo Nell that he took of some sort of a little algae that has a crystal structure inside. And they are very nice and often used to demonstrate resolution, because the little parts of this structure here is very close to each other. It's very close to the limit of the resolution of the microscope. So This is a bright field image, and you can see the object is dark on a bright background. So this is a bright field image. In bright field images, the light goes through the sample. So the sample must be transparent or thin. And it's available on wide field microscopes. So wide field means those with a camera and no scanner. And it's available also on confocal systems. So we can do transmitted light imaging, also often called bright field imaging, but yeah, we can do that on a wide field system and also on confocal system. Yeah. So I remind you, wide field means that the illumination is not focused. Confocal, the illumination is a spot. And then in wide field, it's a defocused, it's a wide illumination. So you can say that transmitted light imaging or bright field is a little bit like when you look at the stars on during the day, you're not going to see them. It's a low contrast technique. So that is one of the limitations of transmitted light imaging is the contrast. But the contrast can be enhanced using some optical elements. And so this is the case for differential interference contrast, that you can see here, which gives like some sort of a 3D effect, and also phase contrast. And so these are images of the same objects taken with the 3 different techniques. And then there is another one called dark field, which will appear a bit later. When we increase the contrast, we can have a high optical resolution. So you see these lines, they are very close to each other, but we can see them. We have a very high optical resolution, but a low contrast and no molecular specificity. So we are not labeling in any way. So we cannot label one particular molecule. But the contrast can also be enhanced using colored dyes. For example, in here we have chromogenic dyes, and that you know are used in the clinic, and these are very good. But the problem anyway is that you can label antibodies, so you have a high molecular specificity, but you have a low resolution because the way they work is that they precipitate. So it makes a big blob at the place where you have one molecule. And on top of that, you have very few colors to choose from, and sometimes it's difficult to differentiate the colors. And you require a color camera for this to image this type of samples. So they are not fluorescent. This is transmitted light imaging or bright field. And so what are the applications? It's excellent. Transmitted light imaging is excellent to put fluorescence in the context of an unlabeled tissue or a cell. In fluorescence, you only see what you're labeling. So you will be able to see the context with transmitted light imaging. So that is very good. It's also used a lot in the clinic. It's a rather cheap technique. So these are all label-free. This is all label-free microscopy, whereas this one is labeled. An extra technique that to contrast transmitted light imaging is called dark field. It's used a lot in material imaging. And so all this is some type of bright field, and this is the reason why very often bright field and transmitted light imaging are used to say the same thing. But there is an extra technique called dark field, okay? So in this course, we're going to talk about transmitted light imaging to talk about all these techniques. So transmitted light imaging is very useful to see the context of things, and you can see here how the nuclei maybe are labeled, but the cells are not labeled, and we can see them with great details when the way they move. And then there's a lot more fantastic stuff going on with AI, where we can pseudo label. Basically, AI can find the different areas or something like this and pretend that they are labeled. So they can make a channel like this. It looks like the nuclei is labeled, but actually it was found with AI. So that's a big revival of transmitted light imaging. It's very low invasion, so it doesn't hurt the sample. Right, so now we are going back to our light path. And what is the light path for transmitted light? So we have, again, the light source, and the light path ends with the sensor. So the sensor can be the eye, camera, or detector. And in between, we have the optical elements like lenses, filters, and sample. So this is an inverted microscope because the objectives are below the stage. And the light path is very simply mostly sample, objective, and sensor. So this is what we have. That's the sample is here on the stage. They'll go through the objective, and then it goes through maybe some filters here. And then we go to either the sensor, which would be here. Currently, there is no sensor here. There would be a camera sticking out, or to another sensor, which is the eyes. So where does the light come from? So the light source, so the excitation, if you want to say light, is going to come. It's not excitation. The light source is going to be on the other side of the sample, because the light in transmitted light imaging is transmission, right? The light has to go through the sample. So if we are collecting the light here, then the light source has to be on the other side of the sample. So the light pass from the excitation light pass is light source to sample. So let's have a look. In transmitted light imaging, the light goes through the sample. The sample absorbs some of the light, and then the rest of the light that the sample does not absorb, is transmitted and imaged. A little bit like, if you remember, yesterday I spoke about the sea, and then when the sunlight is reflected on the sea, we can see all the colors, but when the light goes through the water and then it comes out, then the water has absorbed a lot of things. It's only what comes out that we can see. So it's the same thing for transmitted light imaging. Only the things that have not been absorbed by the sample can be imaged or seen on the other side. So where is the light source? In this case, it is here. So we have the sample. This is the way we are imaging. So the light source has to be on the other side. So the light source and the sensor are always on opposite sides of the sample. It's logical. And the light goes through the condenser. So this is what the condenser is used for. I'm just having a quick look at my timing. Yes. I'm good. So the light always goes through the condenser. So this is what the role of the condenser is in transmitted light imaging. So in most cases, the light source is a halogen lamp. It can be sometimes a white light LED. So you guys will have to figure out what your light source for transmitted light imaging is. So if you see a halogen lamp somewhere, then you will know it's the light source. And the light source is always opposite the objective, so you know where to look as well when you look at your microscope. There's only one exception for this, and again, it is the same exception. So it's when we acquire images of transmitted light with a single point confocal. So in that case, it's a little bit different, and that is because the light source is the lasers. So we use the same light source for fluorescence and for transmitted light on single point confocals. And so in that case, it comes from below. So then it comes from below, it goes through the objective, it goes through the sample, because it's transmitted light, right? So the light goes through the sample, and then it goes through the condenser. And so in that case, we're going to have a sensor up here. Okay, so this is the exception where we are going to have the sensor not on the same side as the objectives. It's when we are on the point confocal, single point confocal, and we are imaging transmitted light. So this is all these are things that you will have to do on your systems. Do you have any idea on your systems where people who use a single point confocal, do you know what the sensor for transmitted light it's called? What's the name of the sensor for transmitted light? Anyone images transmitted light on their point confocal? Here, you can raise your hand. No transmitted light imaging? You guys are missing out, it's very useful. There, we have someone in the audience who is using transmitted light. Well, this is going to stop because you will have to image with transmitted light on your microscope. So that's okay. So on the point confocal, the detector for imaging transmitted light is often called a TPMT, and T is for transmitted. And PMT is a type of sensor, so transmitted light PMT. TPMT, so if you look at your microscope and you see somewhere, it's written often on the sensor. So if you see TPMT, you know that this is the sensor for transmitted light. Okay. Okay, so yes, so in that case we have the sensor opposite the objective, which is the exception. But in any case, in transmission, when we look or image transmitted light, anyway, the light always goes through the sample. That's why it's called transmitted light. Okay, so we have these two light paths. This is for this one is for the single point confocal, and this is for all of the microscopes. The light source is here, and then we have the sensors. And then for the single point confocal, it's the opposite. So now we have a little zoom poll, and let's see here. Let me see. We do that. Oh yeah, here. Polls here. Okay. So I have a little question for you. Click. In your microscope image transmitted light. Okay.
Sylvie, it says hosts and panelists can't vote, so I think we can't vote, but maybe other attendees can vote.
The panelists cannot vote?
No, I also cannot vote.
Ha! That's special. Okay.
Mm,
All right. Well, well, we have a lot of people who can image transmitted light. I think all microscopes can image transmitted light, so that was good. And right, well, this is I will think about that one for next year. I give my other question anyway, if that works. Yes. Is this one? Yes, that one. No. Okay, I somehow cannot put the other question. I don't know why. Here, Anton, that's why. Ah, all right, here, boom. Have you ever imaged transmitted light?
Yes.
Why the transmitted light image? Okay, we do have something. We do have something. It's actually very common. Once you know how to do it, first it's very easy, especially on a point confocal, because you already use the lasers. All you need to do is turn on the detector, so you don't add any light. So we're going to do that, and then you will know. Well, thank you, people in the attendees. So now we're going to do the a little quiz, and so I think that is also part of the quiz I have. Yes, here. There. So if I go to this guy here, and the next quiz is this one. So using your favorite microscope, you can image transmitted light. And you can see that the light comes from the top of the microscope, so it reaches the sample from above. Where is the sensor?
it should be from below.
Below.
So was that?
It should be from below.
So someone says below. Anybody else? What do you guys think? There was someone else who was talking?
Yeah, I think that is below too.
I think that is below as well. Okay, yes, good, it's below. And that is because it's transmitted light. We have the light coming from the top of the microscope, so definitely the sensor must be below. Okay, so that's a very simple one. Thank you. Okay, so now we're going to talk about fluorescence. So the first bit here was about transmitted light, but now we talk about fluorescence. So fluorescence is the opposite. We have bright objects on a dark background, right? In transmitted light, we had the other way around. So bright object on a dark background, and that looks good. So we have two types of light involved in fluorescence microscopy. There is the excitation light, and that is very strong. It comes from a lamp or an LED, like a mercury lamp, an LED, lasers, and it goes to the sample. But we also have another light, which is the emission light, and that is very weak, so really seriously much weaker than the excitation light. And that comes from the excited sample, and it goes to the sensor. So in this one, you can say this is the excitation light path, and this one is the emission light path. So the sample in fluorescence microscopy acts like a light source. So fluorescence microscopy is available on wide field systems with a camera and no scanner. It's available on confocal systems and on light sheet systems. And in our field of research, it's used extensively. It's also used in the clinic, but less. The great advantage of fluorescence microscopy is the contrast. We have a very high contrast, and it's the same thing as looking at the stars during the night. So we see them because of the high contrast, because of the dark background. So the contrast can be further enhanced using colors. So we have access to many different colors. And when we image fluorescence microscopy, we mostly use black and white sensors. So then we have to separate the colors using filters, and that's what we did yesterday. So I hope that you're comfortable with all this. There are many applications, but we have a very high molecular specificity. We can use antibodies against a protein with we can do two antibodies against the same protein with two different conformations. So this is really high molecular specificity. We have a high resolution. There's no such thing as this precipitate that happens with colored dyes. And we have a high contrast because the background is black. Whereas in transmitted light, I remind you, we have a low contrast. We have a high molecular specificity or a high resolution. So we cannot have both together. But we have a low contrast anyway in transmitted light. In fluorescence, very high contrast. So now if we look at the light path for fluorescence, so we have the light source, and then we have again the light path starts with the light source, always ends with the sensor. And then we have in between the optical elements. And the sensor can also be the eyes, the camera, or detector. And the optical elements is the same thing, lenses, filters, sample, etc. So everything including the sample. So what is always true with fluorescence microscopy is that the light source delivers excitation light, and it can be different things. It could be a mercury lamp, it could be LEDs, or it could be lasers. Okay, so this is the type of light sources that we have. I remind you that for transmitted light microscopy, we had a halogen lamp. The halogen lamps are for transmitted light microscopy. Mercury lamps, LEDs or lasers are for fluorescence. The excitation light, so let's say we have the light source here. The excitation light is delivered through the objective, always, okay? That means that the light source and the objective are on the same side as the stage. And then the sample is going to emit light in all directions. So now we have a different light source. In most cases in fluorescence microscopy, only the emitted light going back into the objective is detected. So we're going to have light coming from the sample in all directions, but we can only detect what comes into back into the objective. Okay? This means that in fluorescence microscopy, both the excitation and the emission lights go through the objective. Okay? Very unlike transmitted light microscopy. So both of them, excitation and emission, go through the objective. And that means that we do not use at all the other side of the microscope. So the other side of the microscope is only used for transmitted light imaging. So you can do this little test when you go to your microscope and you take an image of your sample, of your fluorescent sample, and then you open the arm of the microscope. If your microscope allows, not all microscope can do that. If you open the arm of the microscope, or even if you put your hand above the stage to block the light, and you take another fluorescent image, nothing will change. So the top of the opposite side, the side that is opposite the objectives, is not used at all in fluorescence microscopy. So in most cases in fluorescence microscopy, both excitation and emission lights go through the objective. Okay? And one more thing that is always the case is the lasers are never used when looking in the eyepiece because it's dangerous. And so they're blocked basically. There is a way to automatically block it when when the light is going through the eyepiece. So therefore that means that on point confocals, for example, you're using lasers to image, but you must have another light source to look in the eyepiece. So most of the time, or not most of the time, always on point confocals, you are going to have different light sources for fluorescence. The lasers are used to image, and then you're going to have an LED or a mercury lamp to actually look in the eyepiece. So we will have to find all these elements on your microscope. Okay, so don't be surprised if you have an LED or a mercury lamp on the system that uses lasers. They are there just for you to look at fluorescence in the eyepiece. So, if we compare a little bit the light path for wide field and confocal systems, this is a little bit the summary that I've told you of what I've told you so far. So this is true for all wide field and confocal systems. So confocal, regardless of this multi-point or single point confocal. Okay. This bit bothers me. Yeah. We have transmission microscopy and we have fluorescence microscopy. And we have upright and inverted systems. Okay, so here we have our little sample is a fish, and we have our objective. So this is inverted, the microscope, the objective is below the sample. And then we can have upright, in which case the objective is above the sample. The condenser is always on the other side compared to the objective. So here we go, we have the condenser here and we have the condenser here. This is for transmission microscopy. But in fluorescence, we do not use the condenser. So even if we have it, we don't use it, okay? But it's anyway always on the other side. The sensors are on the objective side, right? So this is like most of the case. There are a couple of exceptions. So here we go, we have the sensor for on the same side as the objective. And then when we image transmitted light, the light source is opposite because we have to use transmission. So the sensor and the light source are always opposite each other. And in fluorescence, the light source is on the same side as the objective. So both the excitation and the emission go through the objective. Here we go. So in fluorescence microscopy, we have the sensor and the light source on the same side. And there's only one exception. This is when we are imaging transmitted light on a single point confocal. And that is because the light source used, the light source that is used for transmitted light is also the lasers. So in that case, we place an extra detector on the other side of the objective. Okay, so it's quite sort of streamlined. And again, this applies to all microscopes. So it's sort of universal. Okay, and most of you are going to be in this situation where you have a camera-based system or a single point confocal. This is for transmitted light microscopy, and this is for fluorescence microscopy. So this is basically what you're looking for on your microscope. So now we're going to go to the workshop Zoom, and then we'll have a break. And so we will come back here at 10:30. Okay. So see you guys.